Hi girls, this is the second video on plant reproduction. In this video I want to go through pollination, methods of pollination, fertilisation, seed formation and development, and endospermic and non-endospermic seeds. What's the difference between the two of them? So to get started, what pollination is the movement of the pollen grain from an anther to the stigma to get to the egg so it can fertilise it. So there's two types of pollination. We have self-pollination. This occurs in the same plant. It is essentially inbreeding because the seeds are a lot less sturdy and resilient. They don't last that long. In comparison, where in cross-pollination, it occurs in different plants. So this means that pollen from one plant fertilizes the egg of another. So it has to be the same species of plant though. Cross-pollination is a lot better than self-pollination because it allows for variation to occur. When we talk about methods of pollination, what are we talking about? We're saying, how is that pollen getting from the anther to the stigma? How is it being brought over there? So there's two methods by which this can be done. So we have wind pollination or insect or animal pollination. So the insect or animal is one option and then the wind is the second. So when we talk about wind pollination, we see this in quite boring looking plants, like if we were talking about grasses or cereals or oak, like the example is here. They're quite colourless. They're all green or quite beige looking. The reason for this is because they don't need to be brightly coloured to be pollinated by the wind. Okay, the wind doesn't decide what plant it's going to pollinate based on the colour of the petals. So when we talk about a typical flower of a wind pollinated plant, we see that again there's no brightly coloured petals. We have very light and small and dry pollen grains, so it can be lifted up and carried by the wind. The anthers and the stigmas, they stick out from the actual flower unit. Why do they do that? So the wind can carry the pollen grain off. So the pollen is being brought away from those anthers that stick out of the flower, and then the wind is carrying those pollen grains in the wind, and that pollen is getting stuck in these stigmas here. So the stigmas here, they're large and they're feathery looking, and they stick out from outside of the flower unit so it can trap the pollen grains in it. When we talk about insect or animal pollinated plants, we see a big difference in how these type of flowers look. So if we look at the picture here, we have large, brightly coloured petals to attract insects or animals like birds or bats, okay? We have a nice scent coming from them, again, to attract these animals or insects. We have nectar in these flowers to attract insects and birds and bats, okay? The anthers are inside of the flower, and so are the carpels, the full female reproductive part, are inside of the flower. So the pollen is going to be picked up from the anthers when an animal or an insect comes in to get nectar. So let's say a bee comes in to collect nectar from the inside of the flower. On its way down to get nectar, it brushes against this anther, and the anther has the pollen that sticks to the insect, the bumblebee, okay? So as the bumblebee is carrying this pollen that comes from the anther here, it then travels away and goes to the next flower and comes to the stigma of the next flower. So it comes into this flower, the next flower, to get more nectar, and on its way down it comes into contact with the stigma. And the stigmas, in this case in animal or insect pollination, they're sticky, and they're sticky so they can stick to the pollen that the insect or the animal has brought with it. And that's how insect or animal pollination occurs. Okay, so just to go through those points again, for insect or animal pollination, we have large, brightly coloured petals. There's a nice smell that comes from the flower. There's nectar that's produced from the flower. So nectar is like a sugary syrup that the animals or insects want to pick up. The pollen itself, it's sticky. It's going to stick to the animal, it's going to stick to the insect. And it's heavier than the wind pollinated pollen. The anthers, they're inside the flower and they're a lot smaller. The stigmas, they're inside the flower, they're sticky stigmas, okay? And they're inside of the flowers. Okay, so that's all pollination gone through. So what happens after this point? When we're talking about pollination, why does pollination occur? The pollen needs to get to the carpel to try and fertilise the egg that's inside the carpel. So what is fertilisation again? It's the exact same as in human reproduction. We have the union of the male and the female gamete. So in humans, we would have the sperm fusing with the egg, and they're both going to be haploid to make the diploid zygote. In plants, we have the haploid pollen fusing with the haploid egg cell, and that makes a diploid zygote. Okay, so at this stage, we have the pollen grain has been pollinated, and the pollination has made the pollen grain come to the stigma of this carpel here. So what happens? So if we go through it step by step, because this is a long process as well. So remember when we were talking about the formation of the pollen grain, and at the end stage, inside of the pollen grain, we had two nuclei. We had the tube nuclei, tube nucleus, and the generative nucleus. And around the two of these nuclei, we had the intine layer and the exine, rough or exterior layer. Okay, so the two main things that we need to remember about the pollen grain here is it has two nuclei. It has the tube nucleus and the generative nucleus. So the pollen grain has landed on top of the stigma. The two nuclei are going to separate at this stage, and we have the tube nucleus actually creates this pollen tube that runs through the style of the carpel. Okay, so this tube nucleus is making this tunnel for the generative nucleus to get all the way into the ovule to meet the egg cell down here. So the tube nucleus is making this tunnel, this pollen tube, all the way down here. So the generative nucleus is following it. The generative nucleus is following the tube nucleus. And on its way down, the generative nucleus actually divides again by mitosis. So when it's dividing by mitosis, that means that it was haploid anyway. And if it divides by mitosis, we have two haploid sperm cells now. So what was the generative nucleus has now become two haploid sperm nuclei. So we have, at this stage, we have one tube nucleus that is making the tunnel through the style to get to the ovule, and we have two haploid sperm nuclei that are following the tube nucleus to the ovule. So when the tube nucleus reaches the ovule, it comes down here and it goes through the micropyle, which is the entrance into the ovule. Okay, it comes into the ovule, and as soon as it gets there, it's done its job. It degenerates. We no longer need it for plant reproduction. We still have the two sperm nuclei remaining. So these have an important job here. So there's two sperm nuclei, which will be very different to animal reproduction. One of these sperm nuclei are going to fuse with the egg cell. So that's my zygote made. I have my haploid sperm nucleus, and it has joined or fused with the egg cell inside of my ovule here, okay? And that has made my diploid zygote now. Then I have my second sperm nucleus, and what happens with that? So my second sperm nucleus enters into the ovule, and it is going to fuse with the two polar nuclei that are here. So this will be something new that we haven't heard of before, and it's not something that was involved in human reproduction. So we have our 2 polar nuclei, which each of those are haploid. Okay, so that's that's N plus N. And then we have our haploid polar sperm nucleus that has come in to fuse with the 2 of these. So the 3 of these nuclei are going to fuse together to make a triploid endosperm. So that is my haploid sperm nucleus and my 2 haploid polar nuclei have fused together to make a triploid endosperm. So as a result of the fertilization process here, we have our sperm nucleus number 1 has fused with my egg cell, my egg nucleus, to become my diploid zygote. Then we have sperm nucleus number 2, and that is going to fuse with my 2 polar nuclei to become a triploid endosperm. So you're going to hear that word endosperm a lot now. We're going to be talking about that a lot from this point onwards. So after this point, it is a zygote. What happens to a zygote when it goes through mitosis a few times? It becomes an embryo. So we look down here at a bigger picture, a detailed picture. The whole thing is an ovule, okay? This whole unit is an ovule. Inside of the ovule, we have the outer parts that are still called the integuments. We have the entrance into the actual embryo sac is called the micropyle, the entrance into it. We now have my diploid zygote, and we now have my triploid endosperm inside of this ovule. Now, this continues to develop further when we talk about seed formation. So this is the beginning of my seed because fertilization has actually occurred. After fertilization has occurred and we have the development of the seed further, we have the egg and the sperm have fused together. The egg and the sperm nucleus have fused together, so it's now a fertilized egg or a zygote. As that zygote goes through mitotic divisions, it then becomes an embryo. The integuments that we had on the outside here, they will now harden and become a brown layer that is outside of the seed. So if you think of an avocado stone in the inside of it, and it's brown, okay? But there's a layer on the outside of it that you can almost peel off. That was the integuments that have become the new outside part of the seed, which is the walls of the seed, or another term is the testa. So the testa is the outer part of the seed, the skin of the seed, if you like. The endosperm and what was the nucellus. So if we look back here for a second, just to remind ourselves what the nucellus was. In the actual ovule here, we have our 2 polar nuclei and our egg cell. All of that space that's around that was called the nucellus. Once that endosperm was made by the 2 polar nuclei and the sperm nucleus fusing together, we then had the endosperm. So we had the endosperm inside of the nucellus with the zygote as well inside of the ovule. So the endosperm and the nucellus together, they make a food storage. Okay, the 2 of these together will provide a food storage for the growing embryo inside of the seed. So now let's look at the next stage of how the seed grows. So we had the integuments on the outside. We had the endosperm on the inside and the zygote on the inside. We had all of this part here was the nucellus. From this stage here, it goes to this picture here. So the full thing is called a seed now. It will develop further and continue to be called a seed. Okay. Inside the seed, we have this whole thing here. This whole part here is called the embryo. Okay. This whole part here is the embryo. The embryo consists of now the cotyledon or cotyledons, depending if it's a monocot or a dicot. It may have 1 or 2. And it will have the the plumule and the radicle. So this inner part here is going to be called the plumule and the radicle. You can't identify which part is which right now, but as development continues, the parts will become more identifiable. So the embryo contains the 2 cotyledons or 1 cotyledon, as well as the plumule and the radicle. So the plumule is what's going to become the shoot of the plant. And the radicle, or for radicle and or for root, is going to be what becomes the root system of the plant. We have the endosperm here has become a lot bigger. The nucellus here has become a lot smaller. So the 2 of these are still my food storage or my food reserves for the growing embryo. As it develops further, we have the plumule and the radicle become more identifiable. So the plumule on the top, radicle on the bottom. Plumule again is going to be the shoot system. Radicle is going to be the root system. And this giant thing here that is the cotyledon or cotyledons, the 2 of them. The cotyledons, what happens here is the cotyledon absorbs the food storage that came from the endosperm and the nucellus. So we see the endosperm and the nucellus becoming a lot smaller and the cotyledon becoming a lot bigger. So remember again, what is the testa now was the integuments on the outside. The testa is just the seed covering that's on the outside of the seed. Okay, so just to go over that again, we have what was in the embryo was the cotyledon and the plumule and radicle. Cotyledon is small at this stage. We still have a large endosperm and a smaller nucellus with the integuments on the outside. It then develops further and we have the cotyledon becomes really big, absorbing all of the endosperm, a lot of the endosperm and nucellus. And we have the plumule and the radicle with the testa surrounding everything as a protective layer on the outside. Okay? So moving on from this stage, the last part of today's topic that I want to cover is the differences between endospermic and non-endospermic. So you've heard me talking about the endosperm before. This was the tissue that was made by the polar nuclei fusing with one of the sperm nuclei. The embryo includes the growing plumule and the radicle and the cotyledon or cotyledons inside it. So depending on if it's a monocot or a dicot, okay? So as the cotyledon gets bigger, it absorbs more of the endosperm, as I said before. If it absorbs the full endosperm, that means that there's no endosperm remaining when the seed is fully formed. So if it has no endosperm remaining, it is called a non-endospermic seed. If there is still some endosperm remaining, that means that the cotyledon has not absorbed the full endosperm, then the seed is classed as being endospermic because it has some of the endosperm left. So this links to monocots and dicots like we talked before about plant structure. So when we talk about monocots and dicots, monocots means that we only have one cotyledon, dicots means that we have 2 cotyledons. In general, monocots are endospermic, meaning that they have not gotten rid of all of the endosperm and it still contains some endosperm. So if we think, for example, corn and popcorn, when the popcorn pops, it is the white part of the popcorn that is the endosperm reacting to heat. So we have the corn popping and as a result the endosperm, it pops out, and that is an example of a monocot and that is endospermic as well. If we think of a peanut, when I look inside a peanut has 2 halves, so they are the two cots, the 2 cotyledons, the dicot seed, okay? When I look inside the peanut, there is no difference in the colour of the structure. I can't see different parts to it because there's 2 cotyledons. It means, and there's no endosperm. So all I can see is cotyledon. That's all I see when I look at a peanut. So when we talk about the difference between monocots and dicots, monocots only have one cotyledon. In general, monocots are endospermic, meaning that they still contain some endosperm. Think of the white of a popcorn. When we think of dicots, dicots have two cotyledons. Think of a peanut. And they are, in general, non-endospermic, meaning that they do not have any endosperm remaining. Okay, so that is the difference between endospermic and non-endospermic. Non-endospermic, there's no endosperm left. Endospermic, there is endosperm left in the actual seed. Okay, so that's the video finished for today, and I hope it was helpful. In tomorrow's, I'm going to go through fruit formation, dispersal methods, the dormancy of the seed, and germination methods as well. Okay, thanks girls, I hope it was helpful.